Okay, hi all. This is the third revision area of B2, third and last, inheritance, variation and evolution. So, how did we get to be like this? Why do we all have similar features and yet differences? And there's certain things about us that makes us human, very different from a fish. But there's certain characteristics that makes a fish, and similarly with plants, dogs, flies. Every living organism is a certain way because of their genes, because of their genetics. And this is really a study about how does that genetics work at a basic level. Obviously it's developed a lot more at A level and a lot more in degree level. And also what leads to the fact that everybody has a different height. People have different facial characteristics, different hair colours, if they have hair, different arm lengths, different coloured skins. So all of these things that, although we have the same genes as each other to be a human, within genes, these instructions that we inherit and pass down, and they change over time with evolution, within genes we have something called variation. And that's a real strength. So sometimes strength might give you an advantage. Sometimes smartness, cleverness might give you an advantage. Sometimes with animals it might be the colouring of your skin that gives you an advantage over others. But because of variation, only the best survive. And that's the basis for when we move on to evolution. So to start with genetics, we've got these two words at the top. Asexual reproduction and sexual reproduction. So asexual reproduction, where we have here an organism, simple, one-celled big, and this organism grows and develops, and then it copies itself, then it splits into two, and then they grow and develop and become adult organisms, and then so on and so on, and then they can grow and split again and again. It's called asexual, as in not sexual, and there is one parent. So there's only one parent. What that means is the daughter cells, these two here, those cells will be exact copies of the original cell. So it leads to something called clones, exact genetic copies. And it also leads to little variation. Now the opposite of asexual reproduction is where you have two parents. So here we have a mom and a dad, and they are both providing through meiosis, and we'll come on to that. They're both providing, the dad is providing some genes, the mom is providing some genes in the nucleus of the egg. They come together at fertilization and they form this new individual at fertilization, which can then grow into a new adult. And this is a completely original mix of the genes. Hence why brothers and sisters, siblings, are not identical to each other, but often they have lots of features in common, depending upon what genes they inherited. And sexual reproduction involves two parents. With the two parents you get lots of variation, and with lots of variation we have quite a lot of change over time, or evolution, but as I say, we'll come on to that a bit later. So just a quick summary of asexual and sexual. Sexual, it's this mix of genes. It allows us to mix up our genes and come up with new individuals. You will probably have some of your parents' features or some of your grandparents' features, but actually you're not identical to them. And to make that happen, we need specialised sex cells. And specialised sex cells are called gametes. So we get greater variation, but it's a lot slower than just doing the quick asexual copying yourself, splitting, copying, splitting, copying, splitting. Okay, remember, you're doing a mind map, so pause whenever you need to, and obviously add details. Saw some of the mind maps from last week. Some people have then gone through their exercise book from year 10, or from when they studied it, or gone through bite size and added more detail in as well as they read more and more about a subject. And that can, the mind maps can improve over the next few months. So when you studied B1, which is probably a long time ago now, in Biology 1, it covers mitosis. And mitosis is when you grow. Mitosis would be growth. And even at my age, although I'm not growing anymore, what my body has to be able to do is it has to be able to repair and replace cells that are damaged. So what happens is a cell copies all of the DNA, all the chromosomes. So then we get all the copies, and then it splits into two identical cells. So if I draw two chromosomes in here, and then this one gets two, because it was all copied, and this one gets two, the original ones, and that gets the copies. And that's how you changed over time. You were once one cell big, and later that day you were 2 cell big, and now you are millions and millions of cells. You've kept copying and copying and copying and growing. That's fine. Every single one of your body cells has these pairs of chromosomes. But when you want to make babies, when we want to have sex, sexual reproduction, when we want to pass the genes on, what happens then is we go from these pairs of chromosomes up here, and the first stage is very similar. They are copied. And then it splits, very similar, but then it splits again. So instead of ending up with 2 cells, like we do over here, we end up with 4 cells. So what we say is, for humans for example, in a normal body cell, you have 23 pairs of chromosomes. And these copy here, and they make 23 pairs in this cell, and 23 pairs in this cell. But then they split. So they split the first time here, they split again. When they split again, you get 23 single chromosomes. Now, these sex cells, these gametes here, it might be sperm, it might be eggs. But these cells are only giving half the information, half of the DNA to make a new person. And that's why the sperm and the egg carry half the information each. If it's a plant, it's the pollen and the ova, they carry half the information each. You're quite often asked the difference between meiosis, used to make gametes, sex cells with half the information, or mitosis, used for growth and repair. You might be asked what the differences are between those, just as you might be asked what the difference is between asexual, with one parent copying, and sexual, with 2 parents and half the DNA. So this diagram I like for meiosis because it also shows that actually at one stage here, when the first split happens, so here the original cell, then you copy all the chromosomes. All the chromosomes are copied, so this is one copy and that's the other copy of this chromosome. Once you've copied them, they then split. But look what's happened. A little bit of the DNA from this one has swapped with this one. So actually information moves across some of the chromosomes. And that's why you can be a mixture of your grandparents' and parents' DNA, because some of it's swapped over at the stage in meiosis. And then these cells at the end, they've only got half that information to make a new person. A person requires, in this diagram, 4 chromosomes. This one's only got 2. So this might be an egg, and then it needs another 2 from the sperm. So I've already used the words chromosomes and genes and stuff, but actually you need to understand the structure of DNA. So a chromosome, and that's why the words are in red, remember, these are key words that you have to use in your answers. You need to be able to understand what they are. A chromosome is a great big long length of DNA, and when I say long, I mean extremely long, loads and loads and loads and loads and loads and loads of genes in it. So a chromosome is a long length of DNA made up of lots of genes. A gene then, if a chromosome is made of lots of genes, a gene is, for example here, I could say that this section is a gene. This section, that's one instruction. That might be the instruction for your eye colour. That might be the instruction for how long your humerus, the bone in the top half of your arm, grows. That might be the instruction for how long your eyelashes grow. That might be the instruction on how you make the enzyme insulin, which you need to control sugar level. So each instruction itself is called a gene. Sometimes people can have an instruction that went wrong, so they might be missing a gene and they might have a genetic disease or a genetic condition. But each instruction, and then this next section, might be a different gene. And there might be some bits here. This bit in the middle isn't part of the genes, so we could say it's non-coding. It's part of the genetic code, but it's just like a space between two words. So that's chromosome done, that's gene done. DNA itself is made out of 4 chemicals called nucleotides, which obviously we get from our food, just like we get our carbohydrates and fats and proteins. And the 4 chemicals are called adenine, thymine, cytosine and guanine. And most people know them by their letters, A, T, C and G. The code very often doesn't go wrong because A can only ever join with T and C can only ever join with G. So if you look here, every time you see an A, the green ones, there will always be a T next to it. Oh, there's a T. Oh, that must be an A. There's a A. That must be a T. So actually the genetic code is quite simple. So the nucleotides, the order they're in, code for different proteins, and that's the genetic code. The idea of a double helix, well, that's probably the easiest bit on page. A helix is a spiral shape. And if you look here, look at the 2 blue lines. There's a helix and there's a helix, and they wind around each other with the chemicals going in between. So here's 2 chemicals, nucleotides. Change colour pen, probably easier to see, or we should go with that one. So here's 2 nucleotides joined together by this section of the helix and that. That's why it's a double helix. It's 2 spirals with the nucleotides joining them together. And then we've got the term genome, and we talk about, for example, the human genome. Again, it's in red, it's a key word, it's a word you're expected to know, it's a word that you want to be comfortable using. Human genome is all the genes, all the genetic code of a whole person. So we've mapped the human genome, we know every single gene. Took years and years to do at many universities, all working together on the same project, but we've mapped the whole human genome. But when we talk about the genome of a fish, it's all of the genes to make that fish. So it's not just how many chromosomes you've got, it's not just certain genes, every single gene to make you is your genome. So the genome, by definition, is all the genetic instructions to make an individual organism. So then down this page, and I didn't want to do millions of slides because this is revision. This isn't trying to teach you everything again. It's just covering it quickly. In terms of size, there's you, or actually it looks more like me. We are made out of millions of cells. Each cell has a nucleus. Inside the nucleus we have 23 pairs of chromosomes. Keep saying that because you need to know the numbers. We have 23 pairs of chromosomes. Every cell in your body does, apart from sperm and eggs. And the chromosomes are made of DNA. So actually some of these words on the right hand side we've already covered. So we've already covered what a chromosome is. It's a long length of DNA made up of thousands of genes. And we covered what a gene was. A gene was a small section of DNA coding for one instruction. And we've also covered what a gamete was. About 3 slides ago, gametes are known as sex cells. They're special cells just for sexual reproduction. And the ones we've mentioned so far, in animals generally it tends to be a sperm and an egg. Egg are sometimes called ova from the ovaries. And in a plant it tends to be pollen is the male sex cell and ova or eggs are the female. So there's only really the sperm, the eggs and the pollen. Now the other words in here may sound strange, may look strange, but to try and remind you what they are, we just need to go through them over the next couple of slides. So, as I said, we've done the sex cells, we've done the great big long lengths of DNA in a nucleus, we've done the individual instructions. The next one, allele. An allele is here. So what you will have is you'll have a gene from your mom, and actually I'm better off putting you will have a pair of genes. Your pair of genes, you will have got one instruction from your mom and one from your dad. These are the different alleles available for eye colour, for example. So maybe you received the brown allele, maybe you received the blue allele, maybe you got 2 copies of the brown, so you actually got from your mom a brown allele gene, and from your dad you also got brown. So on your chromosomes you've got 2 instructions to have brown eyes. Another person might have got brown gene from their mom, but then from the sperm of their dad they might have got a blue gene. So then they've got 2 different ones. That doesn't mean they have one brown eye and one blue eye. What tends to happen is that some genes are dominant. Dominant means they are the more powerful gene. So even though this person here on their chromosomes will have brown and blue genes that they can pass on to their children, actually what would they have? Well, if brown eye colour is dominant over blue, so what would they have? Well, they would look like brown-eyed. That's how they would look. That, how you look, is called your phenotype. So, if you're this person and you received a brown gene from one parent, a blue gene from the other parent, brown is dominant over blue, therefore you are brown-eyed. You look brown-eyed. The physical appearance of that inheritance is brown-eyed. However, they have a brown gene and they have a blue gene. This is what actual genes you have, the genotype. So phenotype, what you physically look like, genotype, what actual genes you have. That person there has brown eyes. That person there has brown eyes. They have the same phenotype. but the genotypes are different because two brown genes and a brown and a blue. So actually what they pass on to their children may well be different. And then obviously you might have needed to pause on there for notes. I deliberately put the page again because I knew I'd run out of text. So, sex cells, long length of DNA, section of DNA, different types of the same gene, allele, brown allele, blue allele, emerald allele. A more powerful gene is called the dominant one, and a less powerful one is recessive. So in our last example, dominant gene, brown, recessive gene, blue. Then we went on to what you physically look like is your phenotype, but the genes you have are your genotype. So we've only got 2 words left. And hopefully all these words are going onto your mind map with a bit of an explanation and some drawings or pictures or something to help you remember what they mean. And dominant and recessive tend to be quite easy. People forget allele quite a lot. Homo means same. Hetero means different. So if I wrote this in English rather than Latin, I would write same genes, and that would be different genes. So if you think about the person I just did, and we use letters for these, we don't tend to use whole words like I've been doing so far. So what I might say is, I'm going to call the brown gene a capital B because it's dominant, and I'm going to call the blue gene a small b because it's recessive. The first person we looked at on the previous slide was they got a brown gene from both parents. They would be 2 dominant genes. They are homozygous. They have the same 2 genes. The second person we dealt with, they inherited a brown gene from one parent and a blue gene from the other. So that's why we use the word heterozygous. They have 2 different genes. Now there is a third one that we didn't discuss. The third one would be, what if you received a blue one from your mom and a blue one from your dad? Well, you'd have blue eyes, but that again is homozygous. So how do we tell them apart? We've got a homozygous here and a homozygous here, and it's really easy. That's homozygous with 2 of the same genes, but they are dominant, so we call it homozygous dominant. And these 2, where there's 2 genes but they're both the small ones, they're homozygous recessive. Right, pause if you need to, or otherwise, let's move on. So here's an example. Here, why have we used a capital G for green? Well, we've used that because that must be the dominant gene. And you want to get used to using these words for any cross at all they give you. Here, we've got a yellow one against the green, and they've used the letter G. So therefore I know that's the weaker gene, the recessive one. The dominant would show. So if you get a green and a yellow, you're going to be green. This parent over here started off with 2 dominant ones. So when let's say that it made some pollen, all of them will carry a G. So what I do then is, that G from the pollen could end up with that egg from the female flower. Well, obviously these are peas. Or it could end up with the other one. So actually what we end up here with is, we've ended up with 4 individuals, all got exactly the same. They've all got a green dominant gene, and they've all got a yellow recessive gene. So what would they look like? Well, that would say that if you took a green parent and a yellow parent, all the children end up as being green, because they all get the dominant gene. Doesn't always work out that way, but these diagrams are for you to work out. Based upon any information they give you, you can work out, as long as you know the genes of the parents here and here, you can just add up your rows and columns, and then you find the four, and there's always four possible offspring. Okay? Here's the different possible examples. This one's really obvious. If this parent has the two capitals, the two dominant genes and the two dominant, and what this one is for one of Mendel, we'll come back to him in a minute, one of Mendel's first experiments. With the pea plants, just like those green and yellow ones, he used capital T for tall, and you have to use the same letter so it makes sense, and he used a small t for dwarf pea plants. Why did he use a capital T for tall? Because tall was dominant, meaning dwarf was recessive. So if this parent has two dominants and this parent has two dominants, all the children end up tall. That makes sense, hopefully. Here we have a heterozygous parent. Now what do I mean by heterozygous? The parent had a tall gene and a dwarf gene. So the parent themselves, what do they look like? They look tall, but they carried this gene as well. So they looked tall, but their genotype was they were heterozygous. This parent is identical, they're heterozygous. So when you add up your rows and columns, capital T and capital T, this one is a tall plant. This one was given a dwarf gene and a tall gene. It's a tall. I'm going to write tall for the first one, tall for the second. This one, it got a tall gene and it got a dwarf gene. Still going to be tall. This one received a dwarf gene and a dwarf gene. It's going to be a dwarf pea plant. So even though both parents were tall, they got a dwarf plant in some of their peas off the pod, some of their children. So we say here there are 3 tall for 1 dwarf. That's a ratio. For every 3 tall there'll be 1 dwarf. It's a 3-to-1 ratio in the offspring. And offspring is just a word for the next generation. Rather than calling them children, you don't really say that 2 pea plants had sex and then made children, because not many people think of peas as being children. But actually, offspring is a good word to use for the next generation. Here, one of the parents has both the dominant genes. One is heterozygous. All of them end up being tall. And exactly the same here. If both parents are dwarf, all the children will be dwarfs. What you hopefully will be able to see that here, for example, we had a tall parent and a tall parent, but this dwarf came out of it. And sometimes things skip a generation. So you might carry genes that you don't show yourself, but you might pass on to your children from your parents or grandparents. The person who worked all this out, and the person who is known now as one of the most famous scientists ever, is Gregor Mendel. And it's called Mendelian genetics. He was an 18th century monk. He was a keen gardener, a scientist, but he wasn't really known as a scientist because he had this religious position. He did loads of experiments breeding between different plants, mainly pea plants. He's the one who worked out that if you mix that green and yellow, more of them turn out green, so green must be dominant. He's known as the father of genetics. He's the one responsible for us studying this today. Without any of the equipment and scientific advances we've got today, he worked out basically how genes were passed on from one generation to the next, to the next, to the next, and how they could lead to variation. So in terms of influential scientists, he was a genius. Different example. Here you've got the dominant purple plant. Here you've got a recessive white plant, small peas. Then you've got this plant here that has, and this is where this is supposed to be 2 capital P's like this. This is supposed to be little peas, and this one is supposed to be a big pea and a little pea because they've got those genes. If you put 2 of these together, that's what they're doing here. A capital P and a small p. a capital P and a small p. P's a really bad letter. Think about it a minute ago, we were using T and T. They're a great letter, just like B and B. And they're a great letter because the capital version is very different from the small case. P is a bad letter to use for any diagrams. You don't use P, you don't use S, you don't use O. If you're ever stuck for what letter to use, just use the letter A. It's really obvious which one's the dominant and which one's the recessive. So in this case, even though the parents here are both purple, you can still get a white flower off it. And then one that gets named, the one you don't have to know about the peas and the flowers, you do have to know about the theory of it, and you do have to be able to use those diagrams. This is sex inheritance. Why are you a male or why are you a female? And the simple answer is one pair of chromosomes is mostly responsible for your gender or your sex biologically, male or female. If you received an X chromosome from one of your parents and a Y chromosome from the other, you ended up being a male. Whereas if you received an X chromosome from your mom and an X chromosome from your dad, you ended up being female. You might get asked to explain why half of all births are female and half of all births are male. And it's very simple. This child has got X's from its mom, and this child down the bottom has got an X from its mom. Because in every single egg from a female there is an X chromosome, or there's a pair of them, so every egg has one X chromosome. However, because males are made of one X and one Y, if the sperm that reached to make you, that reached the egg, if it contained the X, it was mixed with your mom's X and you ended up as a girl. Or it reached the other X and you ended up as a girl. However, half of the sperm made contain a Y. So therefore, if it was this one that reached the egg, you ended up as a boy. So the difference biologically is just that girls have two X chromosomes and males have an X and a Y. But that shows you how we can actually explain why are half of births female and half male. Now, as said before, sometimes genes go wrong. And so we end up with things called genetic conditions or genetic diseases. And you only have to know two examples. One of them is a dominant condition. Now, if it severely affected your chance of life, by evolution it would have been got rid of. But the one we need to know about is called polydactyl. And what it means is, poly means many, and dactyl, well, pentadactyl is this, it's limb. So you've got too many limbs. And actually what it means is 1, 2, 3, 4, 5, 6. This person is polydactyl. They have 6 fingers. It happens in loads of different states. If you look on Google, you can find that most of the pictures that come up, polydactyl is much more common in cats than it is in people. But actually, you can have people with 7 toes. You can have people with a thumb, with a small thumb going off the side. But polydactyl is quite a common condition. I know I've taught students in the past, and I've known people who have an extra digit on one finger or one toe, or on both hands or both toes. If it's a dominant trait, it's a more powerful one. So let's use A for polydactyl, and let's lose that for the normal, just 5 fingers and toes, the recessive one. So this person here was an affected male. So this is a man and he's affected. So he's definitely got a dominant polydactyl gene. This one, all these black ones are affected, so that one's got a black, they've got the dominant, that one's got the dominant. Now, what do I put next to this? Well, if all their children, because this shows now the next generation, they had 3 children, child 1, child 2 and child 3. If all 3 children had been affected, this one hasn't been, then I'd know to put 2 dominant genes there. So because that one is white, I know that this one is a small a and a small a. So they must have got the small recessive gene from here. And this parent was there. So now what I can do is, I can put my genetic genotypes on here. So this heterozygous polydactyl person married and had children with this homozygous recessive female. Of their children, 2 of them ended up affected. One was an affected female, number 2, and one was an affected male, number 3. Number 3 got married to somebody else who was unaffected. And they had 2 children, child 1 and child 2. That child received the polydactyl gene and that child didn't. So just because a parent has it doesn't mean you'll get it passed on. But in this case, there's a 50% chance that it would be passed on. It's not a dangerous condition. Some people choose to have an operation just for purely cosmetic reasons to remove it, so that they're not teased for it as a child, or because unfortunately people are a bit horrible with that. Or some people choose to leave it alone because it's completely natural. There's no right, there's no wrong. It's the way they were made. And then the second genetic disease is one that is called, that you will have studied, cystic fibrosis. And cystic fibrosis is a recessive disease. So here, the capital dominant would be your normal gene, and here would be your cystic fibrosis gene. So actually, this person won't have it, this person won't have it, but they could pass it to their children, and this person, the rarer one, would have it. So it's a rarer condition because it's a recessive disease. So you've got one example that you had to know for a dominant disease, that was polydactyl, and now you've got one example, cystic fibrosis. What does cystic fibrosis actually mean for you in your everyday life? It means that your pancreas here gets blocked with mucus, and your airwaves, your lungs, get blocked with mucus. If you've got lots of mucus blocking the airways and blocking the pancreas, it can affect your ability to digest food. So you might not grow as fast, you might not get all the nutrients you want, you might be a bit weaker. And then you've got the other factor here, you're going to have a lot more infections in your lungs possibly, and you're not going to get much oxygen, so you might easily get out of breath. Now, 30-40 years ago, cystic fibrosis was seen as a really serious condition that would very drastically alter your lifespan. Nowadays it's seen much more as a treatable condition, and so for example somebody with cystic fibrosis may take enzyme tablets because their pancreas isn't releasing enough enzymes, so at every meal they take some enzyme tablets, and that can help with the pancreas problem. They might also take steroids to clear up the infections in the lungs, and they might be on a steroid inhaler to open up the airways. So there's lots of treatments now that make it much more a livable with condition. Still serious, still life changing, but a lot more treatments available now to deal with cystic fibrosis. And then we're on to the last part. Well, second to last part. But by now, you've hopefully done about 40 minutes and you're thinking, when will this ever end? I would suggest you take a 5 or 10 minute break. And maybe you come back to this bit a bit fresh. And now you're back from your break, so we'll carry on. So, how have organisms changed over time and evolved? So you'll quite often see graphics like this, and the fact that we evolved from an ape. Well, maybe, maybe not. At the same time as this animal has been changing over time into us, this animal has also been changing over time into other things. And our closest known relative is a chimpanzee. And when I say our closest known relative, it's not because my uncle is a chimpanzee. What I mean by that is when we look at the DNA of a chimpanzee and we look at the DNA of a human, they are very, 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 very similar. The most similar animal to us is a chimpanzee. We're also very similar to orangutans. So they're our second closest related animal. We haven't got so much in common with a cat. We've got some genes in common with a cat, but not many. But what we say this here, at some time in the past, millions of years ago, hundreds of thousands of years ago, there's a common ancestor. So it's not that we came from chimpanzees, but us and chimpanzees both came and evolved from an animal millions of years ago. So it evolved in different ways and changed and turned into new species. We can't breed with the chimpanzees. That's why that's one species and humans are a different species. Sometimes there's different breeds of dogs, but they're the same species because they can still breed together and make crossbreeds or hybrids. And when we look over time at how animals have changed, one of the best evidences for it is the fossil record. And here is the fossilised bones from 50 million years ago of the common ancestor of animals like a horse and a zebra and stuff. And this Eohippus, 50 million years ago from fossils. And if you look at the bone structure, over millions of years, it changes. And it changes again, and it changes again. And it's gone from having these flat padded feet to basically running on one toe, which is a horse's hoof. And it's gone from this little creature, about 30 centimetres big, as long as your ruler, to an animal that's taller than we are. Well, taller than you are anyway. And so we can look over the fossilised size of the heads and the bone lengths and how it's changed over time, and we can see how this little creature has evolved and changed over time to suit its environment. And that's what we call evolution. So in evolution, what we're looking at is only the best survive. And this is Darwin's work, alongside a Scottish scientist called Wallace. And Darwin and Wallace wrote to each other, and they went and looked at some things, and then they put some ideas together, and they both came out with very similar ideas at the same time, but they'd actually been collaborating on it and talking to each other. They came out with the idea that only the best survive, therefore only strong genes are passed on, and therefore animals become better adapted. And they use words like animals were fitter, as a better fit for the environment. So animals become better adapted, they become fitter, and they change. And they change to suit their environment. It might be that you become better and better camouflaged. The ones who aren't better camouflaged die. The ones who are better camouflaged live. The fast rabbits who can escape from all the predators live. The slow rabbits who get caught die. And that's why every single generation animals get fitter. And the theory was survival of the fittest. And they're words from Darwin himself. Survival of the fittest, only the best survive, the weak die out, the weak get caught, the weak don't get to breed or make babies. So survival of the fittest. And in exactly the same thing, by a different term, again, from Darwin and Wallace, they called it natural selection. So actually, I know when I teach it, I teach about great sultans, and I teach about great big fields full of rabbits. Which rabbits will survive this winter? Well, if it's a really cold winter, then your furriest, fattest rabbits might survive. If it's a mild winter, maybe that's not so important, and maybe the rabbits with the biggest ears will survive. But they'll adapt to their environment. And each generation, there are tiny little changes in the genes, because only the best ones get passed on. Therefore, when we look over millions of years, we can get big changes. But evolution takes time. It takes millions of years. It's a long-term process. You don't see evolution in your lifetime. You see it by looking at the historical record or the fossil record. Which brings me on to the second thing. You need a good understanding of how fossils are made. So, in terms of fossils being made, first thing you need an organism to be dead. But you don't want it to all rot away. If you are dead, if it is buried, usually underwater, in sediment, it might not get a chance to rot away. And therefore, as the sediment is changed over millions of years into rock, it can be fossilized in the rock. And you'll need to add a bit more about this in terms of sometimes we do find fossilized actual bones themselves. And sometimes we find minerals that have replaced the bones as they've slowly rotted away. And sometimes that's number one, types of fossils. Number two. Number three, sometimes we just find imprints. So sometimes the fossils we find are just like an imprint of a paw print or a leg print of a dinosaur. And then over time, archaeologists and amateur archaeologists may find those fossils as they become uncovered at the surface, or they might deliberately go looking and digging for them. So, the animal dies and the body sinks to the bottom of water, starts to rot, gets covered with mud, covered in sediments. The sediments harden into rock. As the bones decay, minerals replace the bones, and therefore we end up with fossilized creatures coming to the surface and fossilized bones of dinosaurs from hundreds of millions of years. And I just said that evolution was always called natural selection, but then you've got to look at the alternative, and that is artificial selection. And artificial selection is where not nature chooses natural selection, it's not the survival of the fittest, it is we choose. So why are cows not very clever? Because we have chosen to breed. And what do we want with our cows? Well, we want them to be docile. We don't want them trying to escape, coming up with clever plans. We don't want them being vicious. We want docile cows. We want cows that grow quickly. We want cows that produce lots of milk. So which ones have we chosen to grow? If I wanted to make people taller, what would I do for the next 1,000 years? Well, what I'd do is I'd take tall people and I would let them breed with other tall people, and their children most likely would end up taller. And every single generation, if I did the same again and again and again and again, I only bred tall people, people would get taller and taller and taller and taller. And that's how we've made cows. We've made cows by over thousands of years of farming. We've only bred the ones with the best temperament and the best milk. We've bred chickens that produce lots of meat and grow much faster. The chickens that we have now for chicken farms and eggs and stuff are much, much bigger than wild chickens. So that's artificial selection compared with natural. We choose in artificial. And then this is the last area now. So hopefully you had your break 10 minutes ago and we're close. Because we've mapped the genome, because we know where some genes are, well, and there's quite a lot of controversy over this because it depends how far we go, but But that means potentially we could remove genes and we could replace genes. We could move a gene from this organism, just a random organism to another random organism. We could move a gene from that one to that one and we could change it. And this is called genetic engineering, where we, right, that's a silly way of spelling it. That might be better. Genetic engineering, where we deliberately change the genetic structure of an organism. Looks really complicated, but in terms of the basics, here is a human cell with its 23 pairs of chromosomes. Here we've taken out one gene from it. So we've only borrowed one of your cells and we've taken this gene from that one cell. No effect on you at all. Here is a bacteria that grows really, really quickly. And we take out some of the plasmid, the DNA from the bacteria, and we cut it open with an enzyme. And then what we've done is we've put that gene in here. So we've given that gene to the bacteria and put it back into a bacteria. The bacteria now thinks, well, it doesn't think because it's a bacteria. All of these bacteria now, we let it carry on asexual reproduction. And this bacteria now produces more bacteria that have the insulin gene and the insulin gene, and so on and so on. And then we end up with millions of bacteria that every day will be producing insulin. So this is an example of where we have used genetic engineering. We have used genetic engineering in order to produce the hormone insulin so that diabetics can inject it. With antibiotics, when we talk about survival of the fittest and mutations and changes in genes, an antibiotic is a chemical we know that can kill bacteria. When we talk about antibiotic resistance, well, over time, bacteria, these simple little organisms with their little plasmids and strands of DNA, they can change and alter and mutate. What that means is they could become immune to antibiotics. Now, I've deliberately rushed through that last bit because I wanted to get to this. So, we started with a picture of a fly and a fish and a person, and actually there are hundreds of thousands of living organisms on the planet. So how do we organise them? And that is what this is about. These are all apes. We know that this one and this one had a common ancestor here. Some of them have developed over time into this and some into this. Some of these ones have changed into two different species. How close are we to an orangutan? Well, us and orangutans had a common ancestor here. And this tree is showing time over millions of years. So this creature that lived here millions and millions of years ago, some of its offspring have developed over millions of years into these different species at the top. All of these different species come from this original species down here. And that's the theory. So how do we group them all? Well, here, for example, are just the primates. So what we've got here is an order like primates. All these creatures have something in common. They're all monkey and ape-like. Then we've got a suborder, a smaller group with the humans in it. Then the humans and apes. Then the humans and then that was the humans, apes and tarsiers and monkeys. Then just the humans and the apes. Humans and great apes. Then we've got just the humans, including the archaic humans that went extinct. And then we've got modern humans. And it's called binomial naming. We are known as Homo sapiens. And that tells us what genus we belong to, which is Homo, and what species, Sapiens. So you are a Homo sapien. There's a couple of other examples. This down here is Canis lupus. Canis lupus. Genus, Canis, dog-like. Lupus, wolf. Like Lupin. But you'd know that if you watch Harry Potter. Or some of the Twilight things. Or some of the other werewolf stuff. So, a Swedish scientist called Linnaeus came up with this classification. Again, 1800s. Where he'd have all of the animals all grouped together, and he called that level of organisation a kingdom. And by animals, I mean everything that is multicellular and not a plant or a fungus. So, butterfly, birds, lobsters, foxes, dogs, bears, every animal at all is in this kingdom. But within that, you then split it into things that didn't have a backbone and things that did. So the ones that are here, all the animals with backbones. And there's 5 main groups in there. You've got your reptiles, amphibians, mammals, fish, birds. And then you've got a class. So we are in the class called mammals. We're not in the same order. Of the mammals, it then came down to carnivores. So mammals that just eat meat. And these were the Canidae, canines, and then the genus. So a wolf is a Canis lupus. It belongs in this family. It belongs in this order of meat eaters. It's a mammal because it's hairy and got warm blood. So that's where we're in common with it. We're in there as well. It's the same as us. We're in the same phylum. It's got a backbone in there with fish and birds and everything. And then it's in the animal kingdom. So it very much goes from there are hundreds and thousands of animals, and at each level you get less and less. And this is Linnaeus's classification system. I know people I teach, lots of them have got a mnemonic, such as kill penguins 'cause overweight fish get stuck. I know is one of them. Different example. Sorry, I'd already mentioned Linnaeus on that previous slide, and I'd already mentioned binomial. Binomial means 2 names. That's how we're known. That's why we're a Homo sapien, and this one is genus Canis, species Lupus. So we call a wolf Canis lupus. This is Leo the lion. Its name is Panthera leo. A lion, it's in the animal kingdom. It has a spine. It's a mammal because it's warm-blooded with hair. It does eat meat, so it's in that carnivore. So a lion is in the same group, the same order as a wolf. But where the family was Canidae, this is Felidae, the cat-like family. So a lion is in a different family, a different genus, and it is a different species to a wolf. But the name, the binomial name, Panthera leo. There he is. Panthero leo, binomial name. And then after Linnaeus, and this is the very last slide, after Linnaeus, a lot more discoveries were made from the 18th century when Linnaeus came up with his classification system. And when Linnaeus was looking at it, he was actually looking at this. He was actually looking at single-celled organisms, plants, animal, fungi. He was looking at these kingdoms. And that's why he had the animal kingdom, and he had the plant kingdom, and then we had the fungus kingdom and the protist kingdom for a long time. And we hadn't really dealt with all of these living things here. Lots of them hadn't been discovered, they were too small, and lots of them hadn't been discovered because we hadn't been to loads of the places where they live. And then Carl Woese, much more recently, in the 1980s and 1990s, he added another layer. So above kingdom, he added a domain. So the only main change to Linnaeus' system is we now have 3 domains. First domain is bacteria. These are our everyday bacteria, the ones that cause diseases, the ones that are healthy for our skin and our intestines, loads and loads of bacteria in loads of different places all around the world. But then he said that these ones genetically were so different from them. And these were archaic bacteria. They're known as archaea. So bacteria as one domain, archaea as a second domain. And what are they like? Well, extreme salt condition, extreme heat conditions. They're quite often known as extremophiles. They're very primitive life forms that haven't changed much in millions and millions of years, and they're very, very different to other bacteria, as different as bacteria are from us. And then he had this domain, eukarya, which means these ones all have a nucleus. So all the living things that Linnaeus was doing was basically splitting up this domain. So Carl Woese added a different layer on top of kingdoms, 3 domains, bacteria, archaea and eukaryotes. And now we are finished. And you finished B2. And well done, look forward to seeing your mind maps.